So first of all, I really want to thank all the families that have donated blood. It was amazing. It was much, much more than we thought, and many more, of course, more blood and more people. Let's start like that, a quantitative assessment of that. And we'll give you numbers how many people donated after we really looked through things, probably by tomorrow. So more to come, but I really, really also want to thank the doctor who did that with his wife, Dr. Rampf, right? Because we never met before, and that's one of the amazing things. It's just they're friends of Simi, and she talked them into it, and so we're really, really grateful that you really pushed through. And we also have a little present for you. So this is a picture of neurons with PURA cells. It's explained on the back.
Yes.
And it's signed by all the scientists from our team. Perhaps something for your practice you could put up. There's a surprise. I would like to take it away from you right away, because you're taking off, right, for vacation.
Yeah.
So perhaps if all the families who donated blood and want to also sign here, please do so. And Simi would bring it to your sports event, right?
Great.
So, you know, won't be any clapping. It will be like that. Doesn't mean you're crazy. It means appreciation. So thank you so much.
You're welcome.
Thank you.
Thank you.
And have a safe weekend trip, and hope to see you sometime again with less stress. Yes, thank you so much.
So,
if you want to look at it, you can just drop it off in the bag.
Yes.
Okay, thank you so much. Yes. Okay, so let's go to science again, and this is fundamental science. So it's at the other end when you come from the gene review. So what I want to talk about is we want to understand what's really going on in PURA syndrome in a mechanistic way. Because when you look at it, PURA syndrome is a neurodevelopmental disorder. At least this is how it is described. But nobody really proved that it's the development that is changed. And you know, we're clearly convinced that we have to know what is happening before you can apply the right therapeutic approach. This is different to some other groups, that's fine. We take the approach to be probably slower, but we think we believe this is the right way. And future will tell, you know, if that is true. So this is a very old drawing from Conrad Wellington, where he proposed a model how development works, so differentiation of cells. You all know in the beginning you have stem cells, right? So this would be a totipotent stem cell. That means it's a cell that become everything, even embryos. And his idea was that you have a landscape, and that the stem cell is a ball that rolls down the landscape. And depending on where it rolls, you know, it becomes a different cell type. So if you and that means if it rolls down the ball, it differentiates, so it loses its potential to become everything, but they can then become all kinds of cells. So, you know, if you roll down a little bit, you end up with so-called pluripotent stem cells. They can become a lot of things, but not embryos anymore. And then if it further goes down, you know, it can become skin cell, or what do we have here? Nerve cell, or blood cell. So depending on the path it takes, it becomes something different. And the direction it takes, this is governed by the gene expression profile. So there is a gene expression profile turned on if the cell should become a skin cell. And of course, if we talk about PURA and we think that something goes wrong with the development, it is important to know where that happens. And this is what I mean with a fundamental understanding. You know, if something happens up here, it has very different implications on which therapeutic approach we should take than if it's happening down here. And that's why we do the very careful step-by-step approach. Now, I want to introduce you to a magician, a science magician. Here he is, Yamanaka. He's an amazing guy. He won the Nobel Prize for his discovery that you can turn the process around and you can make cells forget what they were and become pluripotent stem cells again. And this is a wonderful research tool, because with this you can take cells from people, make them pluripotent, and then just repeat the development and, you know, check what's going wrong. So this is what we do, and others also. And you heard that, you know, other people also use these iPS cells. That's the abbreviation, APS, induced pluripotent stem cells. So we can take cells, skin biopsies from person patients. We can grow the skin cells, and then we apply the so-called Yamanaka factors. This is the cocktail that makes the cells forget what they were, and hey, they become stem cells and are happy to become whatever we tell them. So, we can then tell them to roll down as a ball in the one direction and along the landscape or into another direction. And this is really, really helpful because we could use it to understand abnormalities in development, in cellular functions, in molecular functions. And of course, the goal is to establish treatment strategies. That's our declared goal. So what we've been doing is to obtain samples, skin samples from PURA syndrome patients. We reprogrammed them and made them iPS cells, and you see there's a plus/minus. This means one copy is intact, the other one is broken. This is the typical genotype of PURA syndrome patients. But you can also take another approach, which is take these cells from healthy people and then use CRISPR-Cas gene scissor to destroy both copies. So you see PURA minus/minus. And when you think about, what model would you use to study PURA syndrome? I guess your first guess would be, take the upper one, the patient cells, right? But we discussed this a lot, and we felt that, we're not analyzing humans directly, but cells derived from humans, and it might not be completely the same as in the patients when we use this as a model. So it might be that some weaker phenotypes, some weaker abnormalities, they would be hidden in an ocean of signals, because we look at thousands of genes at the same time. So we decided that in order not to miss anything, we will start with looking at cells that have completely lost the PURA function, because then we get the maximum effect, and then we understand, what all the abnormalities are. And then we can go to the stem cells from PURA syndrome patients and really look at, which ones are relevant. So that's the approach we took. And I will talk about what we do with these full knockout cells. So you see, we can differentiate them into neurons in petri dishes, but you can also differentiate them into something that we call brain organoids. So this is something, it's a complex process where you let them grow in 3 dimensions, and the cells start to self-organize, like in our brain, and they talk to each other and establish most of the major layers in our brain, the different types of neurons and so on. So you can very nicely use it to understand if the self-organization, the development of these neurons happen correctly. And so this is what we did. And this is work done by Sabrina Bacher, former PhD student. She left more than a year ago, but was a wonderful study by her. All right, so what can you use these brain organoids for? It's kind of the gold standard these days, because you can use it to study all kinds of things: toxicology, brain tumors, neurodegeneration, and so on. What is most interesting for us? Neurodevelopment, PURA syndrome, and of course you can study things that happen before the birth, which you can of course not analyze in patients, right? So it's a unique window of opportunity to really look into these early stages. And that's what we did. So we generated these organoids, and these are light microscopic pictures. So you have here the healthy cells, and then in comparison, the ones lacking PURA. And that's important. You always need a control, a proper control in research. You always need something to compare to. And when you look at that, you don't see any major growth difference. You see here 0.5 millimeter in size, so these are a couple of millimeters. So I'm not sure, but I guess the ones who visit the Helmholtz tomorrow will be able to see them. Yes, I get a nodding there. And they're like little white styrofoam balls, but they have living neurons in there. And the experts here see that there is no statistically significant difference in the size, first of all. So no dramatic effect on the size. Second thing, we thought, okay, perhaps something inside the organoids is different. So we cut through them and then stained them for different things. And what you see is the green stuff, these are neurons. The red stuff is PURA, which is present, of course, in the healthy cells, but not in the PURA knockout, so the cells that lack PURA. And you also see that there are different layers of tissue. This is the differentiation, you know. Our cortex has an outer layer with neurons, and then if you move towards inner parts, you have different structures. But from this we conclude, well, we did not observe any obvious differences. And we really tried very hard because we thought, you know, we have to find something because PURA patients, you know, they have some differences. But we were not convinced, and we thought, okay, no, this is not the right approach. So what can you do? You need a more detailed analysis of PURA lacking brain organoids. And this brings me again to the structure. So this is a schematic drawing of the different layers. You know, you saw at the outer side, which was kind of stained in, can I go back perhaps? Yeah, which stained in green here. These are the neurons. And so, yeah, they're red here, well, not ideal, but then this is the outer layer. And then you have all kinds of different cell types, lots of different subtypes of neurons. And down here you have the ventricular zone. The experts know ventricles are like fluid-filled areas in the brain. So it really recapitulates a lot of the things that we see in the brain. So what do? We thought that perhaps, you know, because there's not so much of visible differences, it's only a few cells that are affected. Perhaps, you know, only a certain cell type is perhaps not working properly, or is not functioning, or is not present enough. And perhaps some cell types become more and get more and some get less. Very difficult to study, but there's a technique that is really helpful there, and this is called single-cell RNA sequencing. And this is a beautiful work we've been doing with Anna Dück, great collaboration. She's sitting there in the back. We're really grateful for the collaboration. And I'll show you now what this means. I try to explain it, and I hope you understand it, what we do with this. So, analogy. You have a fruit bowl, you're at breakfast with your spouse, and you realize, oh, I forgot my cell phone. You go back to the bedroom, come back, and you have the suspicion your spouse ate one of these fruit pieces. How do you go about, how do you find out? If you're German, you know, we'll be very careful, count the things. And this is exactly what you do with the cells. So we go, and by the way, it's a very nice book by Ursus Verli, who's not a German, he's a Swiss guy, they're even worse than us. So what we do is we take these brain organoids, and they have between 8 to 10,000 cells, and we dissociate them. That means we separate each cell so they become an independently floating cell in a soup of, you know, liquid. And you have all the different cell types mixed here. And then we put labels to a different label to each of these cells. So about 8 to 10,000 labels, we call that barcoding. And then we sequence them, and we can sequence each individual cell. And then we can really sort these cells and map them. And the representation, that's a scientific representation, is this. So each dot here is a single cell, and we sort them by their similarity. So going back to the fruit bowl, you know, this would be bananas, we would have kiwi down here, apples, so we sort them this way, right? And obviously there's breeds between banana and kiwi, they're kind of intermediates. But that's how we do it. So let's look at the results from the PURA organoids. So this is what we get when we analyze it, and you can see we color-coded it. And here you see the different cell types, and the experts know that there are, you know, all kinds of different neurons we could identify. And you see up here you have really neurons, and here's something that is called progenitor cells. So cells that are in the landscape more up, you know, they haven't rolled down the differentiation landscape completely. You find them below here. And what we found in the end, I'll just summarize that, don't show any original data, is that we have an imbalance between excitatory and inhibitory neurons. So excitatory neurons are the ones that fire, send signals, and inhibitory neurons say, hey, stop, not too much, right? And it's the balance between the active and the ones that put the brake on that provides the right balance. And it's a little bit like, the analogy comes in a second. And we realized that this imbalance happens early on in the progenitor cell already. And so you can compare this imbalance somehow to a soccer game. You know, that's the topic of the month, obviously. Just imagine you have a football team and you have offense and defense. If you have too much of offense or a weak defense, not good. And you don't have a good offense but only good defense, you might be better off. But in the end, if you want to have a really strong team, it doesn't work. And it's the same with PURA. So we have probably too less defense and too much offense. And that is, I think, the main conclusion. So the question is, can we transfer that knowledge from the organoids to the human development? I told you that this happens early on and so on, but that's a very general, broad statement. It's not very scientific. So we looked in the literature if there is reports which cell type occurs during early prenatal development. And there is a study. We were happy that we don't have anything to do with it, because what they did was taking aborted embryos, and they did the same thing, single-cell RNA sequencing, and then looked at which time point after conception, and this is the maturation state, which cell type appears. And you see here at the very beginning these radial glial cells, and these are the cells that already make the mistake. So this tells us that obviously when we take this analogy seriously in the first trimester, it is likely that the abnormalities occur. So this is, of course, not a direct evidence, but this is, I think, the best scientific assessment we have to date. There's no other data out there, so I think this is what the most likely scenario is. All right, implications for patients with PURA syndrome. We have to be careful not to overstate, because we talk about little cell balls in a dish, right? We don't talk about patients. But it is likely that we have prenatal developmental deficits, and that we have an imbalance of the different types of neurons. And this is a possible explanation for epilepsy, because this has been described in other disorders, that this misbalance, having too many firing neurons, is connected, may be connected to overshooting of the brain signaling. And to follow up on that, we teamed up with Chris Group and an Australian team, and because they have a mouse model and they study exactly this topic. So we hope that with a very nice ping pong, we learn much more than if each of us would push forward. What else? Because of the likely prenatal onset, treatment options after birth may be limited. And I think this is the most important thing to us. It seems that a cure is not possible, because diagnosis is always happening after birth, and some of the changes have happened already. But that does not mean that there is therapeutic success after birth. So I would color that in a color I call hopeful green, because I think we can't say how limited it is or how much benefit there will be. We just have to find out. We have to develop treatment options that work after birth. It's unlikely that they would completely cure the kids, very unlikely, I think. But there might be a good window of opportunity to have significant benefit. And this is, I think, in my eyes, the most realistic a range of expectation we should have. And the next presentation by Marta Somorai, she'll be catching up on that, because she will talk about the experiences she made as a clinician on related disorders. One more thing before you come. Limitations of the study, also something very important in science. So the study was performed with a complete deletion of both copies of PURA, right? This is not PURA syndrome. So the PURA syndrome patients only have one broken copy. So it's likely that the symptoms are weaker. We're not sure, we don't think that the onset, the time of the abnormalities might dramatically change, but obviously the symptoms are weaker. Second thing, cerebral organoids are not an entire organism. We don't have blood vessels, we don't have systemic things. That's why we team up with people like Chris's group, who really check what's happening in an entire organism. And with this, I'm done. I'd like to thank everybody. We're in contact with, I think, all the major patient-related organizations. We'd really like to thank the PURA Syndrome Foundation Germany, who supports us, and individual grants from people, funding agencies. And would like to highlight again Sabrina and Anne for their wonderful work. Thank you.